You will see the objectives for our innate immunity content listed. Today is the second mini lecture, and the content of this lecture will largely focus on the recognition of pathogens by cells of the innate immune system and the induction of inflammatory cytokines. Cells of the innate immune system, including macrophages and neutrophils, have pattern recognition receptors that trigger cytokine production. So here I've shown an epithelial barrier and an uninflamed connective tissue you will find present all the time, tissue-resident macrophages and dendritic cells. These cells can respond if there is a disruption or abrasion within this barrier that allows for the entry of bacteria. And the reason they can respond is they express receptors that are specific for broadly expressed components of these bacteria. And these are often referred to as pattern recognition receptors. They allow them to recognize the bacteria and initiate an inflammatory cytokine response. Other cell types, such as dendritic cells within these tissue, also express these toll-like receptors or these pattern recognition receptors, but they migrate to lymph nodes when they recognize bacteria and they activate T cells. So we will focus on dendritic cells more in the adaptive immunity lectures. So what are these patterns that are recognized by these pattern recognition receptors? Well, they're often referred to as pathogen-associated molecular patterns, or PAMPs, and include things like lipopolysaccharide or lipoproteins or lipoteichoic acid, peptidoglycan, flagellin, fungal sugars, bacterial DNA structures, and viral nucleic acid structures such as double-stranded RNA or uncapped messenger RNA. The main family of these pattern recognition receptors is referred to as the toll-like receptors. And toll-like receptors are expressed at the cell surface. They recognize these various bacterial products. There's toll-like receptors that are expressed within endosomes that recognize bacterial nucleic acid. There are other families of pattern recognition receptors that are also expressed in the cytoplasm. And these include the Rig-like and Nod-like receptors. The Rig-like receptors are particularly important as they are able to recognize viral nucleic acid structures and are probably the most important family for detecting viral infection. This is a picture of some of the various family members of the toll-like receptor family, and as you can see, some of these family members are expressed at the surface and some in the endosome. You don't really have to memorize all of these and their specificity. We're going to focus on one family member, toll-like receptor 4, which is activated by lipopolysaccharide. So here we have a gram-negative bacteria. Lipopolysaccharides are expressed at higher levels in gram-negative bacteria in their membranes. And lipopolysaccharides are either on the cell surface, but bacteria sometimes secrete these lipopolysaccharides. And these lipopolysaccharides can bind to toll-like receptor 4 on the surface of macrophages or neutrophils in combination with this other receptor CD14. And so this CD14 toll-like receptor 4 complex recognizes these lipopolysaccharides. And when that happens, it induces an inflammatory response. There's an adapter protein called MID88 that associates with these toll-like receptors, and when it becomes activated, it induces a kinase signaling cascade that eventually results in the activation of a transcription factor called NF-kappa B, listed up here. And NF-kappa B induces the production of inflammatory cytokines such as interleukin-1, tumor necrosis factor, interleukin-6, interleukin-8, and interleukin-12. This MID88 is a common signaling adapter for most toll-like receptor pathways. So for immunodeficiency purposes, you really don't see very many severe immunodeficiencies associated with loss of one toll-like receptor or another. But if you lose this adapter, you do experience severe immunodeficiency in your innate response. So what are cytokines? These are small secreted proteins. They mediate both localized and they can mediate systemic communication between cells in the immune system and in other organ systems. They can have immunostimulatory and immunoregulatory functions. You'll see families of interleukins and interferons and colony stimulating factors. They're important in inflammation and adaptive immunity, in cellular trafficking and hematopoiesis. And so cytokines are really the way that immune cells communicate with each other. Here's a macrophage that's secreting a bunch of different cytokines. I've mentioned interleukin 6 and tumor necrosis factor and interleukin 1. CXCL8 is another name for interleukin 8 and interleukin 12. Interleukin 6, TNF, and IL-1, these are all really important in mediating the local and systemic inflammatory response. IL-8 is what is known as a chemokine. We'll talk more about it later, but it helps mediate trafficking of cells to sites of inflammation. And interleukin 12 is a really important cytokine for activating certain cell types, including natural killer cells. But we will talk more about interleukin 12 and its role in the adaptive immune system later on. So what do IL-1, IL-6, and TNF do? Well, all of these cytokines bind to cytokine receptors on phagocytes, which helps induce their activation. IL-1 and TNF are particularly important in activating endothelial cells. This induces vasodilation, vesicles, and increased leukocyte recruitment from the blood. Interleukin 6 is important for inducing the acute phase response, which leads to systemic inflammation induced by certain liver products. Interleukin 1 is really important for induction of fever and is sometimes referred to as an endogenous pyrogen. And tumor necrosis factor is really important as an inducer of septic shock. However, all of these cytokines can participate in all of these processes. So what are some of the local effects of these cytokines? Well, they activate local immune cells, they activate endothelial cells, they induce endothelial or capillary vasodilation, they induce chemotaxis of leukocytes from circulation to sites of inflammation. And they also have a number of key systemic effects that can include inducing the release of neutrophils from the bone marrow, increased hematopoiesis through production of certain cytokines such as granulocyte monocyte colony stimulating factor. There are prolonged systemic effects. So if you have elevated, sustained inflammation, you can have a hard time it can be hard for your hematopoietic system to keep up in producing new neutrophils, and you may see an increase in circulating immature neutrophils, sometimes referred to as bands. As I mentioned, these cytokines can induce the acute phase response, which I'll describe in a moment. They can induce fever and rashes and septic shock. So let's talk about some of these systemic effects of these cytokines, and we'll begin with the acute phase response. This is largely induced by IL-6. Now, IL-6 is produced locally, but within 24 to 48 hours after the initiation of an infection, many of these cytokines enter into the blood and are now available systemically. And when this happens, these cytokines can travel via the blood to the liver and produce the production within the liver of certain proteins that enhance the systemic inflammatory response. These include increased production of complement components, increased production of components that enhance pathogen recognition. This includes mannose binding lectin and C-reactive protein, or CRP. It includes increased production of components of inflammation and hematopoiesis, like cytokines such as GM-CSF, and some other components that are also serum amyloid A and phospholipase A2 that do play a role in inducing inflammatory responses. And lastly, there's certain coagulation factors that are produced, such as fibrinogen and plasminogen. And I do want to mention to you, elevated fibrinogen is detected with an assay called the erythrocyte sedimentation rate. C-reactive protein is often used as an inflammatory marker. And so when you go through your case-based learning modules, you'll come across ESR or CRP tests. These are testing systemic inflammation and are components of this acute phase response from the liver. There are these cytokines also are key in inducing fever, and sometimes are called endogenous pyrogens. IL-1 is particularly good at this. It acts on the hypothalamus to increase the activity of COX-1 and COX-2 enzymes. These enzymes specialize in converting arachidonic acid into prostaglandins, which can induce fever and a number of other systemic effects. And as I mentioned, cytokines can have local effects, and so TNF can have very beneficial local effects, such as activating phagocytes and activating endothelial cells and so forth, as we've mentioned. But it can have very harmful systemic effects if it is present at very high levels for extended periods of time, and it can induce septic shock. And so high levels of systemic TNF induce systemic vasodilation and edema, loss of organ perfusion, and organ failure. Now, I've mentioned cytokines that are largely induced by bacteria. I do want to mention now a cytokine family called type 1 interferons that are induced by viruses. And when I refer to type 1 interferons, I'm referring largely to interferon alpha and interferon beta. And so here's a cell that's infected with viruses. And as we mentioned, there are certain cytoplasmic pattern recognition receptors, such as Rig-like receptors, that can detect viral nucleic acid structures. And when these pattern recognition receptors get activated, they lead to the activation of a class of transcription factors called interferon response factors that in turn induce the production of type 1 interferons, interferon alpha and beta, that are secreted. And so type 1 interferon is often referred to as the antiviral cytokine. When interferon alpha and beta binds to receptors on neighboring cells, it can inhibit viral entry, inhibits viral replication, inhibits protein synthesis, inhibits viral budding. Most cells can produce type 1 interferon, and most cells have receptors for type 1 interferon. And the antiviral effectors that are induced by type 1 interferon have really broad effects in limiting viral propagation. In some cases, it can be viewed as an emergency response that may lead to cell death and prevent viral spread within tissues. And some immune cells, like dendritic cells, are particularly good at making type 1 interferon. And one thing that I should mention is that type 1 interferons, interferon alpha and beta, they activate natural killer cells. And natural killer cells are important effector cells for the antiviral response. Okay. And this is illustrated here. Natural killer cells migrate from blood vessels and patrol tissues. They're activated by interferon alpha and beta, and as well as IL-12. They're a key early source of a cytokine called interferon gamma, which leads to enhanced macrophage activation. This is another one of those cytokines that we'll talk a little bit more about during our adaptive immunity module. Type 1 interferons, or interferon alpha and beta, are important for direct killing of infected cells. Infected cells, or all cell types, but especially infected cells, will express receptors that indicate stress, or ligands that indicate stress. Natural killer cells have activating receptors that detect these ligands. Natural killer cells also have inhibitory receptors that detect the expression of MHC class I. Killing by NK cells of a target is induced because you have an abundance of these stress ligands on the surface and a scarcity of MHC class I on the surface. And this is shown in this picture here. Here we have a natural killer cell and a target cell that is expressing these activating ligands, stress ligands or whatever else, and MHC class I on its surface. And when these inhibitory receptors are engaged by MHC class I, this NK cell is not activated and it does not kill. But infected cells upregulate the expression of these stress ligands and often downregulate the expression of this MHC class I. And this induces the activation of natural killer cells, and now it will kill this infected cell. So here are some of the key points from this mini lecture. You can go through these points as you review this material to make sure that you have fully understood everything that I've talked about today.